Okay, so good day, everyone. So this is the continuation of our lecture, on-site lecture for this week. So we have methods of identification part 2. No, since we were able to cover only until MVIC, no, and there are still some biochemical tests left to be discussed. So eto na yon. Okay, so to continue, we have urea hydrolysis. No, so of course, the enzyme that we want to determine or identify for this test is the enzyme urease, which, if present, will break down urea, which is present in the culture media, into molecules of ammonia, carbon dioxide, and water. Okay. Now, if ammonia is released, this will cause a change. Now, it will trigger the pH indicator phenol red to change the media from pale yellow, no, like this, to dark pink or magenta. Okay, so the usual culture media to be used for this particular test is Christensen's urea agar or Stewart urea broth. Okay, again, the pH indicator is phenol red. Okay. Usually, those that are strongly positive for urea hydrolysis is the ppm, no? Your Proteus, Providentia, and Morganella. Although others can weakly produce urease and may cover the slant portion with magenta or pink color. This would be your Sikis, okay? Bisaya nga Shakis. So you have your Citrobacter, Klebsiella, Enterobacter, Yersinia, and Serratia. Okay. Then, of course, you have the motility test, which we perform on your SIM. Okay, so the SIM is, of course, a culture media that can be used to determine three factors, no? Sulfide production, indole production, and of course, motility. Okay, so your SIM is a semi-solid media, which is, again, perfect for determining if the organism is motile. Okay, so atong bantayan diri is the spread of growth from the point of inoculation. So, for example, itong whitened part here, no, sa second tube, okay, yan ang point of inoculation. Pag nag-spread ang growth or if the entire medium becomes turbid, then the organism is motile. Okay, pag ganito siya, ganyan lang, nandyan lang siya, hindi siya actually motile. Okay, it has to be really cloudy or nag-disperse from the line of inoculation. Okay, para maging motile. Now, most of your Enterobacteriaceae are motile with the exception of Shigella, Klebsiella, and Yersinia. They may be motile at lower temperatures, but at incubation temperatures, they will lose their motility. Okay, so they're considered non-motile in the laboratory assays. Okay. Then we have the decarboxylase test. Okay, so simply, based sa pangalan niya, we're trying to determine if the organisms are able to release the decarboxylase enzyme, which will remove the carboxyl group of amino acids. For this test, we usually use lysine, ornithine, and arginine. These are amino acids. Okay, so if the organism are able to produce the decarboxylase enzyme, the carboxyl group of these amino acids will be removed and converted into amines and carbon dioxide. For lysine, okay, lysine will be reduced into cadaverine lysine, causing a change in color of the medium to alkaline. Ornithine, on the other hand, will be hydrolyzed into putrescine. Okay? Arginine naman, okay, arginine is usually reduced to amines in a roundabout manner. This would require two enzymes actually. So arginine will be acted upon initially by dehydrogenase enzyme, no, into citrulline, and then citrulline will be further reduced into ornithine. So as it becomes ornithine, the decarboxylase enzymes will now act on it, no, to reduce it into putrescine. Okay? So medyo paikot, no, ang kay arginine. Okay? Now decarboxylation usually would occur only in the presence of an acidic environment. Okay? Si decarboxylase enzyme, mamatara na siya kung acidic ang environment. So this test is also a type of indirect carbohydrate fermentation test. Okay? So ang culture media natin dito is molar decarboxylase broth, or actually you can use your LIA for this, no? The molar medium would contain 1% of lysine, 1% of ornithine, and 1% arginine. Okay? And of course, glucose is present. Diba? Nga naman. Kinahanglan ma-ferment ni siya sa organism to produce acids. And when acids are produced, no, that would provide the perfect environment para ma-activate ang decarboxylase enzyme. So, no, ang principle niya, if the organism is able to act on glucose and ferment it, producing acids, initially, the medium will become acidic, turning it from purple to yellow. Okay, so that's the initial color change. Okay, so nagiging yellow. Okay, si Bromcresol purple, ma-trigger siya to change, no, into yellow. Okay, so once yellow na yan, acidic na siya, mamata na si decarboxylase enzyme, and it would start to act on the specific amino acid. Okay, so lysine to cadaverine, ornithine to putrescine, arginine, no, will be acted upon by the dehydrogenase enzyme, and then it would be reduced to citrulline, and citrulline will be further reduced to ornithine, and once ornithine na yan, that's the time decarboxylase enzyme would act on it to reduce it further into putrescine. So si ornithine o si arginine, pareho sila og end result. Okay? So once these amines are produced, no, they will turn the media alkaline. Okay? So si Bromcresol purple, ma-activate na naman to change the color of the medium from yellow to purple. Okay? So purple, yellow, purple ang color reaction ng ating decarboxylase test. Ganon rin naman ang observe natin kay lysine iron agar when we do decarboxylase testing. Okay. So, trivia. Meron tayong triple positive na organism. So, ibig sabihin nun, sa tatlong amino acid, positive siya. Okay? Positive siya. That would be your Pleishomona shigelloides. And meron din naman tayong triple negative. Wala jud siya decarboxylase enzyme. It cannot reduce, no, the amino acids into amine and carbon dioxide. So that's your Panthea agglomerans. Okay? So we have lysine iron agar. As mentioned earlier, this agar is used for decarboxylation. And at the same time, pwede po ni siya gamitin for deamination. Okay? So lysine iron agar would include the amino acid lysine and of course, naagyapan ng glucose. Kaya again, your decarboxylase enzyme would only be activated in the presence of acids. Okay? So ang indicators, we have your hydrogen sulfide indicators, so yung dalawa, and of course the pH indicator Bromcresol purple. So again, if the organism is able to ferment glucose present in the culture media, it would cause the culture media to become acidic, hence ma-trigger ang change, no, from purple to yellow. And then, that would be also the time na magising ni si decarboxylase enzyme to reduce lysine into cadaverine lysine. So when cadaverine lysine is released, no, which is an amine, it would change the color of the media into purple na pod. So ang positive for decarboxylation is purple. Also, no, to note, decarboxylation would occur anaerobically on LIA. Therefore, dito siya ma-detect sa butt portion, no? So yung ating LIA, ito yun, slant and butt siya. So the carboxylation is observed at the bottom because it will occur anaerobically. Okay? Whereas ang deamination, okay? So deamination is triggered in the presence of the aminase enzymes. Okay? It will occur in the presence of the aminase enzyme, and the activity would be aerobic, and so it is usually observed at the slant portion. So kung sa decarboxylation, tatanggalin yung carboxyl group ng amino acid, sa deamination po, tanggalon po ang amine group ng organism, okay, ng amino acid ni organism. Okay? So tatanggalin ni organism yung amine ni lysine. Okay? So also the positive result for if the positive result for decarboxylation is change from purple, yellow, purple, si deamination naman sa slant is a change from purple to red. Okay, so purple to red. Still, no, the reddening is due to the presence of or the release of alkaline material, no, so causing the change in color of the medium. Okay. Also, ang ating, no, similar to TSI, pag may hydrogen sulfide production at makoveran yung buong bat, kagaya nitong tube number 3, okay? So this is also considered positive, okay, for fermentation. Okay? So maunang maano siya, no, ma-produce yung ating, ma-produce yung makoveran rather, yung buong bat. Okay? So positive din yan for decarboxylation kung, no, yung buong bat niya nangitim dahil sa presence ng hydrogen sulfide. So ang ibig pa rin sabihin yan ay nagising nagka-acid yung environment at nagising yung decarboxylase enzymes. So, hindi lang natin makita, okay? Pero ganun ang reaction usually, no? Unlike in TSI, recall, sa TSI, di ba may mga ganito din tayong na-observe na mga hydrogen sulfide production where the entire butt is covered. Pero it is still reported as acid butt, right? So yellow butt gihapon na siya. Kasi in LIA, ang kanyang hydrogen sulfide production will occur in acidic environments. Dito naman kay LIA, okay? So this usually would occur in alkaline environments, okay? So magkaiba sila ng pH requirements sa dalawang culture media natin. So kay TSI, ang hydrogen sulfide production na maramihan would occur in an acidic environment, whereas in LIA, the H2S production, no, mass production ng H2S is usually occurring in alkaline environment. Okay? So ang reporting pud gihapon ani is positive decarboxylation. Okay? Then we have the phenylalanine deaminase test. Okay, so ang ating ginagamit dito is the PEA agar or phenylethylalanine agar, which would contain 0.2% phenylalanine and 10% ferric chloride. Okay, so the enzyme being determined here is phenylalanine deaminase. So if it is present, it would act on phenylalanine to produce phenylpyruvic acid. Now, ang phenylpyruvic acid cannot be seen, no? It cannot be determined kung naan na bagod siya. So for us to be able to visibly, no, see na naa siya, kailangan munang mag-react, no, yung ating na-produce na phenylpyruvic acid sa 10% ferric chloride. So yun yung purpose ni ferric chloride, para mas maging visible yung ating phenylpyruvic acid. Okay? So common itong mga ganito, di ba, in most biochemical tests, kagaya sa indole, so we add the aldehyde reagent para makita si indole. Otherwise, di nato siya ma-determine. Di ba? So ganun din naman sino pa ba? para makita yung acid production, no? Most of the pH indicators, ganun ang kanilang task. Okay? So for phenylalanine deaminase, si 10% ferric chloride is also added to visualize phenylpyruvic acid. So dito, bonggang-bonggang nagpa-positive na naman ang ppm na magkakabarkada. So si Proteus, Providencia, and Morganella are the only genera positive for phenylalanine deaminase. Okay, so ganyan, no? Dark green ang positive color ng ating deamination. Okay, for phenylalanine deaminase. Okay. So next we have the miscellaneous biochemical tests. Okay, ito yung mga, they're not regularly done in routine laboratories. They may be performed on request. Pero as of the moment, I'm not quite sure if they still perform it sa lab. But I do know they have the materials, no, needed to perform it sa laboratory when requested. Okay, so gelatin hydrolysis or the gelatin stub method. So you will be using your nutrient gelatin deep, no? O but lang yan. Okay, but gelatin siya. So parang slightly semi-solid. Okay, so that would contain around 12% gelatin. So humukay siya pero muhigda ragud gyapon kung imo siyang ipaslant. However, kung imo siyang i-refrigerate, mag-gel siya konti. So magporma siya konti. Okay. So this to do no your gelatin hydrolysis, you just stab it. Okay? Remember na wag ipaabot sa dulo, no? So just stab your gelatin and then incubate that for about 2 days. And then prior to reading, imo sana siyang i-refrigerate for at least 30 minutes para ma-set. Okay? Para makita natin yung difference, okay? Kung meron or walang gelatinase enzyme, okay? So after 30 minutes, titignan mo na ngayon immediately, no? Wag mong tagalan kasi baka slightly mag-liquefy din siya. Okay? So immediately after removing from the refrigerator, check mo, okay? Slightly tilt it, no, to see the difference, okay? So kung dili siya mag-tilt, okay, nag-stable siya in that position for at least 1 minute, o di negative yan. Pero kung it would start to tilt like this after a minute, ano siya, yung positive siya for gelatinase enzyme. Now, for this test, it is known that only serratia, no, ang nag-hydrolyze ng gelatin. Okay? Okay, so this is an example of a commercial biochemical method, no, or biochemical test. Nandiyan na lahat ng kailangan natin, guys. So notice those small triangles na nasa baba. Okay, yun yung parang separator. Naka-groups yung mga test natin. So yung first test dyan, that's ONPG, arginine dehydrolase, lysine decarboxylase, your ornithine decarboxylase, ayan, di ba? So nakatatlo-tatlo by threes, I think, yeah. Except for the second set na 2 sets ay tig-dalawa lang. No? Yung sa gelatin at saka sa glucose and your amylose and arabinose. Okay? So this is the API 20E system. Diba? Biomero pa rin to. So ano yan? Parang isang set na ng biochemical reactions. All you need to do is to place the emulsified solution. Yung ano ba yan? Lipat ng lipat. Emulsified solution of your organism. Dito, kasi may mga butas yan dyan. No? So once na nalagyan mo na yan ng iyong inoculum, the next step is to place a layer of mineral oil. No? Mineral oil ang nilalagay to seal. Okay? Para hindi madisturbo yung ating reaction na pwedeng mangyari dun sa ating mini agars, no, sa baba. Ang cute niyan. Tapos i-incubate. Okay? After, edi pwede mo nang tignan, no, kung ano yung mga reactions. And then there's numbers. May numbering yung mga reactions nila. Ganyan. Okay? Tapos, no, titignan mo yung reaction. Ganyan. Isusulat mo. Okay. Tapos i-add mo ngayon yung reaction lang. Di ba sabi ko nga kanina, tig tatlo-tatlo. Okay? So titignan mo ngayon yung reaction. Okay? Per 3. tests. Tapos i-add mo. So 1 plus 2 plus 4. So itong 1 and 2 is based on, meron naman tayong ginagamit na color chart, no, to determine kung anong number yung reaction na nakikita natin dito. So tube number 1, 1 lang yung kanyang reaction. Si number 2, 2. Si number 3, 4 ang kanyang reaction. Ganon. And then i-add mo yun silang tatlo. So 1 plus 2 plus 4, 7. Tapos yung tatlong set na naman, negative lahat, edi 0, 0, 0. O, di 0 yung kanyang total. And so on, no? And then, so meron ka ng code, 7031645. O, pagkatapos nun, i-feed mo yan dun sa machine. Ang galing, di ba? So, meron ka ng organism. Meron tayong mga ganyan. Ngayon, na-upgrade na, meron tayong Vitec. Di ba? So, mas pas-pas. Okay? Then we have serological testing. So as we've mentioned in the class, no, na serological testing is done if we come up with organisms na maraming serovarieties. Or, no, we also do this sa microbio to determine if may tawag nito, may production of antibodies, no? So you could determine titers like your ASO, RPR, and Widal tests. So of course, these are usually in the laboratory, guys. This is done in your serology section. So I believe na-perform nyo na to in your serology laboratory, di ba? So you have your ASO, which is to determine your anti-streptolysin O, your RPR, which is used to determine the presence of your rapid plasma reagent pag merong syphilis ang pasyente, and of course, your Widal test naman for typhoid fever. Okay? So, pero ngayon, yung Widal, kasi parang napakatagal na niya, no? Napakatanda na serological testing. Yan natin mas mga bagong technique, no? Like the Typhi-dot method, na parang na lang siyang yung, lateral flow method na lang. Parang yung mga pregnancy test na lang, isang drop-drop na lang ng serum. Tapos, titingnan mo na lang yung, i-observe mo na lang kung may linya, na lang na siya karon, guys. Okay? So, again, no, very useful ang serological testing and microbiology. Okay? Maraming salamat kay Rebecca Lansfield, di ba? Okay, of course, there are some cases where you could perform animal inoculation, pero we do not do that in routine laboratory settings. No? Wala tayong test na ganyan. Wala kasi tayong maraming space sa actual laboratory. Usually mga reference labs or research laboratories where they keep houses of lab animals. Okay? So ano ba yung mga pwedeng natin, no? Test animals? Usually, yung mga rodents like your mice, no, white mice, yung mga guinea pigs, or in the case of Mycobacterium leprae, pwede naman siya sa armadillos like this. Pero kawawa naman, di ba, no? Your leprae will survive underneath the shells of this armadillo kasi malamig. Diba ayaw na, hindi pwedeng mabuhay si leprae pag 37 degrees, namamatay. So kaya dun siya sa may footpads kasi it's away from the main body. And of course, if you have your PCR, you can always perform molecular techniques, no, for your microbial identification. So one example of molecular assays that we could do to identify your bacteria would be hybridization. So DNA hybridization methods are usually used to identify genomic species. Okay? Pero all of these methods are really time consuming and they are laborious, no? It can only be applied in special situations. So pwede tayong gumamit ng quantitative bacterial dot filter or yung nitrocellulose filter method, yan, no? Pag hybridization. And then of course, yung ating amplification, ito yung trabaho ng PCR talaga. It would amplify the genetic material of the target. So, di ba ganito lang kaliit yung target, hindi mo kailangan marami, so, konti lang. Pero i-amplify yan kasi we make use of templates, di ba? So, madodoble-doble yung number ng ating genetic material na na ipaprocess. So, mas madaling hanapin. Okay? And then of course, we have the sequencing and enzymatic digestion. Ang DNA sequencing is considered as the gold standard for microorganism identification. Okay? And then usually, ang ginagamit nating target na gene, no? Kasi when you do sequencing, there's always a gene target. So 16S ribosomal RNA or 16S rRNA ang ginagamit na gene. Yun yung madalas na target natin for bacteria. And then, medyo mahaba kasi siya. Okay? So, mga around 1,540 plus base pairs ang haba niya. Okay? So kaya ideal siya for most identification. Okay? And then for some, like sa mycology, no, for demataceous fungi, for example, pwede rin naman yung calmodulin genes, no? They could serve as targets for sequencing. Okay? So, for more information on selecting the appropriate database pag nag-sequencing kayo, you can always refer to the Clinical and Laboratory Standards Institute or yung sa CLSI na MM18 document. Okay? So the MM18 document contains the interpretative criteria for identification of bacteria and fungi by targeted DNA sequencing. Meron silang ganun. So again, CLSI document MM18. No? So to choose the specific type of sequencing and the specific type of target gene, okay, for sequencing. Okay? So, recall natin, methods of identification, umpisa yung microscopy, right? Diba? So, microscopy natin. Tapos, mayroong direct or indirect. So, sa atin, we do direct method, specifically differential. So, meron tayong gram staining and acid fast. So, once you have your morphologic data or information, the next step is to culture. So when you talk about culture, syempre merong culture media. And your culture media may be classified according to physical form. So you have your liquid, solid, and semi-solid. According to distribution, you have your plated and your tubed. And sa tubed, meron tayo ng butt and slant. And then according to composition, you have yung tawag nito, natural, synthetic, and yung ating complex media. Okay? So, complex yung natural, parang beef broth, gano'n. No? Pero hindi nga natin alam kung ano yung exact composition. Okay? And then, according to functional type, ito yung marami, right? So, meron tayo ng general purpose media or your basal media. You have your enriched, enrichment, selective, differential, meron tayo nung pang-assay, meron nung pang-enumeration, yun. Okay? And then, of course, for isolation of pure cultures, you have your strict plate, diba? So, using your quadrant streak, you have your pour plate, you have your selective media, and of course, animal inoculation. And then again, of course, if you have your if you come up with species na maraming serotypes, so you do serological testing. And for molecular identification, of course, pwede namang hybridization, mag-amplification, and sequencing. Okay? So that ends our mini discussion to continue what we had discussed, no, on site. So thank you for listening.